Welcome to the Deep Dive, where we unpack complex topics. Today, we're plunging into CRISPR.
Ah, yes, CRISPR.
Clustered, regularly interspaced, short palindromic repeats. It's quite a mouthful.
It is.
But fundamentally, it's this revolutionary gene editing technology. It's actually adapted from a bacterial immune system, which is amazing in itself.
Right.
And it allows for incredibly precise changes to DNA. So our mission today, explore its, surprising origins, the sheer power of its applications, and also the profound ethical tightrope it forces us all to walk. Okay, let's unpack this.
Indeed. What's truly fascinating, I think, is how this completely natural defense mechanism has been sort of repurposed. It gives us this unprecedented, almost surgical control over the very building blocks of life itself.
Here's where it gets really interesting, right? The story of CRISPR, it actually began with scientists just studying bacteria, not human genetics initially.
Basic science.
Exactly. They noticed these peculiar kind of repetitive DNA sequences in bacterial genomes. And it turns out when a virus attacks a bacterium, the bacterium literally saves tiny pieces of that viral DNA.
Like little molecular mugshots.
Yeah, like wanted posters, saved directly into its own genome.
Okay.
Then, if that same virus shows up again, the bacteria use these mugshots along with a protein called Cas9, which acts like a molecular scissors.
Right, a cutting enzyme.
To instantly recognize and just shred the viral DNA. So scientists basically stumbled upon nature's own highly effective immune system.
And scientists, including Jennifer Doudna and Emmanuel Charpentier, who famously won the Nobel Prize for this work.
A huge achievement.
Absolutely. They cleverly adapted this bacterial system. So instead of waiting for a virus, we now design a synthetic guide RNA. You can think of it as a custom GPS for that Cas9 enzyme.
Okay, so it guides the scissors.
Precisely. This guide is programmed to match any specific DNA sequence we want to target in a cell. It leads Cas9 right to that exact spot where it makes a very precise cut in the DNA strand.
And then the cell tries to fix it.
Exactly. The cell's natural repair mechanisms kick in, and that's where the magic happens. We can either trick the cell into deactivating a gene when it repairs, or if we provide a DNA template.
Ah, you can insert something new.
Yes, insert entirely new custom DNA sequences. This remarkable precision and, frankly, the relative ease of use compared to older methods.
Right, older techniques were much clumsier.
Far less efficient, much harder. It really makes CRISPR a game changer.
That's the core mechanism then. And its simplicity is almost deceptive considering its power. The real-world applications are already, astounding. In medicine, it's already being used in human clinical trials for some really devastating genetic diseases, like sickle cell anemia and beta thalassemia. The aim is to correct the actual underlying genetic error.
Which was unthinkable not long ago.
Exactly. And we're also seeing incredible promise in immunotherapy, modifying a patient's own immune cells so they can specifically target and destroy cancer.
Yeah, CAR therapy and beyond. And if we connect this to the bigger picture, its reach extends far beyond just human health. Think about agriculture.
Oh, right, the crops.
Yeah, CRISPR is enabling the creation of crops that are more resistant to drought or pests or disease. This could potentially boost food security and nutrition globally. Imagine, crops needing less water or fewer pesticides.
That has huge implications.
Huge. It's also vital in animal research. Scientists can create very precise models of human diseases in animals for study, which accelerates drug discovery, and even exploring ways to, say, prevent viral outbreaks in livestock.
So what does this all mean then? When we gain this ability to essentially rewrite life's code, with such unprecedented power comes profound controversy.
It does.
A major ethical concern, often dubbed designer babies, centers on this idea of editing human embryos, not just to cure disease, but maybe to enhance traits.
Like intelligence or appearance, yeah.
Which raises significant fears about, exacerbating genetic inequality, creating a sort of gene editing tourism for those who can afford it, and fundamentally altering the human germline changes that would then be passed down through generations.
And that, the germline editing, is a really critical line for many people. But beyond this sort of designer baby debate, there's also the very practical, critical risk of off-target effects.
Meaning it cuts in the wrong place.
Exactly. Where CRISPR might accidentally cut the wrong part of the DNA, causing unintended and potentially harmful genetic changes. This was chillingly brought to the forefront back in 2018.
Ah, the case in China.
Yes, when a Chinese scientist controversially claimed he had edited human embryos to make them HIV resistant. The global scientific community widely condemned this,
Not just for the risks.
not just for the potential off-target risks, which were significant, but because it involved germline editing without robust ethical oversight or, frankly, clear medical necessity at that stage. It led many countries to quickly ban or heavily regulate such procedures.
So the scientific community is working on making it safer.
Oh, absolutely. There's active research to increase CRISPR's precision, reduce those off-target cuts, exploring new versions of the Cas enzyme, better delivery methods. It's a very active field.
This deep dive really shows CRISPR as, well, a double-edged sword. It's a beacon of immense hope for medical breakthroughs, for agricultural advancements. Yet it's fraught with these profound, unavoidable ethical challenges that we as a society are really only just beginning to grapple with.
And for you, the listener, the knowledge of CRISPR's immense power really demands reflection, doesn't it? What responsibilities do you think humanity holds now that we're gaining the ability to rewrite the very code of life itself? And maybe more importantly, what lines should we absolutely never cross?